An international expert panel is calling for an immediate halt to the use of the novel COVID-19 mRNA injections after they discussed in length the scientific evidence of cancer-promoting DNA contamination within both Pfizer and Moderna vials, potentially prompting a series of suspected repercussions that could impact human cell lines for up to 2 generations.
And this is very concerning. So this can pass on to 2 generations in this manner. And it's something that most definitely must be looked at along with the actual integration of the DNA itself.
It has been more than 6 months since your last dose of COVID-19 vaccine or your last infection, your protection from the virus may have waned. Receiving a COVID-19 vaccine dose this fall with an updated formulation is expected to increase individual protection against infection, symptoms, and severe disease.
Over the last year plus, I have brought you various reports underscoring the vast unknowns and the downplaying of risks associated with the novel COVID-19 mRNA injections. Most recently, internationally renowned experts in the fields of immunology, genetics, research sciences, and general practitioners called for an immediate moratorium on the use of novel genetic vaccines, according to a press release issued by the World Council for Health, who hosted the panel. The WCH, in their dedication to health and well-being based on real and true informed consent, hosted a 4-hour-long expert panel on Monday, October the 9th, to discuss the discovery of DNA impurities and the undisclosed presence of a genetic sequence contamination known as the SV40 promoter found within vials of both the Pfizer and Moderna COVID-19 mRNA injections. These shocking discoveries were first documented by genomics expert Kevin McKernan, whose work has since been confirmed and replicated by several international labs.
It is not the full SV40 virus that was seen in the polio vaccines many years ago. However, there is still concern that if this DNA is at high levels inside these vaccines, that promoter could integrate into the genome, and if it integrates in front of an oncogene, then it could promote the expression of an oncogene, let's say. So there's concern of injecting people with high amounts of these SV40 promoters, because if there's any amount of genome integration that occurs, then there's a risk that it will be promoting the expression of genes inappropriately in the genome.
There is no constructive purpose identified for the undeclared SV40 promoter sequence, reads an emailed press release from the WCH. In addition to its cancer risk, SV40 enhances the capacity to incorporate the other foreign genetic material into the recipient's own chromosomes, potentially rendering them, and possibly even their offspring, permanently genetically modified. Toxicologist and molecular biologist Jancy Lindsay stated that this could result in intergenerational effects for up to 2 generations.
My very large concern that these gene therapies will be passed on to our progeny and will contaminate the gene pool. And this is not being investigated at all. Not a single person has investigated sperm or ova to see if these are being genetically integrated. And I have reached out to multiple labs asking them if they would investigate this. We have an in vitro lab that is willing to work with anyone who is willing to test both sperm and ova for integration. Genomic integration could result in cancers rather than just functional integration. In fact, it's unlikely that we will have functional integration into the genome, creating a spike protein, but more likely that you'll have insertional mutagenesis leading to cancer. But there is another mechanism which you can pass on functionally expressed spike protein, and that would be through this extra chromosomal DNA or RNA through sperm-mediated gene transfer. So both of these mechanisms could result in a constitutive expression of the spike protein in some or all cells of the progeny, depending on the way it's inherited. But again, less likely through actual integration into the genome, more likely through this mechanism called sperm-mediated gene transfer. So this is a picture of some sperm that have been transfected with exosomes carrying DNA. And this is courtesy of Dr. Corrado Spadafora, who really was the first to show that sperm-mediated gene transfer was occurring. Through this mechanism, we could be passing on these genetic elements to our children. Sperm-mediated gene transfer shows that it is not required to have stable integration into the genome in order to pass on low copy number, chimeric expression of extra chromosomal DNA expression. And this is very concerning. So this can pass on to 2 generations in this manner. And it's something that most definitely must be looked at along with the actual integration of the DNA itself.
Stressing the importance of informed consent, the release says that it was impossible as the risks of the products have never been formally and transparently addressed by regulators and are not fully known. The failed COVID-19 vaccine campaign has been a biological disaster for the world. Cardiologist Dr. Peter McCullough, who is also the scientific advisor to the WCH, told the panel. With independent assessments such as the above now taking place, dangerous implications discussed by this panel included disease, death, transmission, and intergenerational inheritance. The organization is calling for an immediate halt to the continued use of these novel mRNA products for that very reason. And meanwhile, Canada's regulatory agency continues to endorse the mRNA injections for all Canadians aged 6 months and up. For Rebel News, I'm Tamara Ugolini.
With today's authorization, NACI is reaffirming its guidance on the use of COVID-19 vaccines this fall. NACI recommends authorized age groups get an updated COVID-19 vaccine dose this fall, 6 months after the last vaccine dose or infection. We will have enough supply of the updated COVID-19 vaccines to support immunization programs across Canada.
Someone should be held responsible for the various oversights responsible for the indiscriminate push of these novel injections onto every Canadian. If you agree that Chief Public Health Officer Dr. Theresa Tam has failed to follow the actual science as she continues to endorse these novel injections for anyone aged 6 months and up, then consider signing our petition and supporting our independent journalistic efforts at firetam.com. That website again is firetam.com, and we appreciate your continued support. Thank you very much.